Good morning, everyone, and thanks for joining us for the first quarterly meeting of the Fish Monitoring Work Group. I'm Meg Detloff, and I work for PNAMP. I'll be helping with communications, logistics, and general cat wrangling. First off, I would like to introduce the Fish Monitoring Work Group core team. If you guys want to turn your cameras on and unmute yourselves and give your name and organization and just a little wave so everybody knows who the core team is, that'd be great.
Russell Scranton with BPA.
Nancy Leonard with StreamNet.
Casey Blazner with Oregon Department of Fish and Wildlife.
Marika Dobos with Idaho Department of Fish and Game.
Jen Bayer, USGS PNAMP.
All right. Awesome. Thanks, guys. And the next thing we would like to do is have the participants, well, let me back up here and do some housekeeping. So if you guys are looking at Jen's screen, down at the bottom is the little mute icon that I was talking about. So you guys will want to mute yourselves just so that you're not distracting everybody else from the meeting. You can turn your camera off if you want. You can leave it on, that's fine. The other thing is the little speech bubble box is your chat function. So if you click on that, you'll be able to see the chat and type in the chat. You can type in any questions or whatever comments you may have during the meeting if you don't feel comfortable speaking. So the next thing I'd like to do is to have everybody just click on that chat button and introduce yourself and what organization you're from. That'll help everyone in this meeting connect and be counted. So next slide, Jen. And you guys can just do that while we're kind of going through this. So today's agenda, we're obviously kicking off with the welcome and introductions. Next step will be reviewing the charter. After that, Casey will talk about the work plan, and then we'll jump into the Fish Monitoring Work Group task team updates. And those will be pretty brief. It's just kind of whatever tasks are currently active. We'll kind of give you guys an update on what's going on. And then we'll listen to a tech talk from USGS's Toby Kock. And then we'll wrap up and adjourn and just see how it goes. So I'm going to hand this off to Nancy for the charter.
Okay. Next slide, Jen. So for the charter, we didn't want to go through it page by page today. We figured that'd be a bad way to start off our second quarterly meeting with the Fish Monitoring Work Group. So instead, if you want to take a look at the entire document, you can access it through a link that was included in the email for today's meeting that was sent out by Meg. But for today, we wanted to focus on 3 components of the charter as of most relevant to the folks attending the meeting today. And the first one we want to touch on had to do with the responsibilities of the general member, which is pretty much everyone that's on the call today and others that couldn't join us. So the responsibilities of a general member were described in the charter as basically 3 main things. One was that as a general member, we want your help in identifying topics that can be addressed by a task team under the Fish Monitoring Work Group. So this is similar to what we did during the last quarterly meeting where we all got together, we looked at potential lists of topics, and then we voted to figure out the level of interest and which topic would be worth initiating in the near term. Another responsibility of the general member has to do with helping us identify, topics of interest related to fish monitoring, technology, data management, analysis that we think would be of interest to the broader group. The CAP core team, the Fish Monitoring Work Group core team, we do our job at trying to find potential topics and presenters, but in order to get a nice diversity of topics, we hope that the general members, will speak out and let us know, contact the core team to let us know of potential topics and presenters we should be reaching out to for the quarterly meetings. And PNAP staff will be helping with all the logistics of that. It's more for you bringing forward potential topics. Another aspect related to topics of interest is sometimes, having a guest presenter like we have today with Toby works perfectly for the topic, but other times you may need to flesh out more of a workshop. So if we do encounter that situation, we're hoping that the general members will also help us identify where we can host that workshop. And again, PNAP would be helping facilitate all the logistics around any workshop that we host for the fish monitoring work group. Along the same theme of learning and sharing and expanding our knowledge and exposure to what's happening out there, new technology, new research findings. We've also put together a wiki page on the Fish Monitoring Work Group Teams channel so that you can all provide links to publications, citations to new publications, and we can have an easier way of accessing what each of us identify as a potential interesting new resource that might be of interest to the rest of the group. And the last responsibility, which you're all fulfilling just by being here today, is attending the quarterly meeting. So we expect all the general members to attend our quarterly meetings so that we can get your input on topics, presentation, workshop, and just keep learning from each other. Next slide. The other two sections of the charter we wanted to focus on has to do with the task teams. As you might remember from the last quarterly meeting, once we identify as a group specific tasks that we think are ripe to move forward, these cannot move forward without having a lead and members. So we want to just remind everyone what's in the charter, this new charter related to the responsibilities of the lead or leads and members. The task team lead is a voluntary position. We hope that when a task is identified, there's a lot of excitement about it, a lot of interest for it, that one of the fish monitoring work group general members will step up, just like we're going to be touching base on some of these task teams that have started already later on in today's meeting, that volunteers will step up to say, yes, I'm willing to lead, I'm willing to coordinate, I'm willing to keep momentum going, make sure that the product that is needed will be developed in a collaborative way through a task team. As a task team lead, you're mainly overseeing the task team. PNAP staff, especially Meg, helps you with the logistics of it, getting oriented to teams, getting your members access to teams. But you're basically the driving force to make sure that the task team as itself has a clear work plan, understands what the product goals are, and keeps the core team up to date on what progress is being made. Just similar to how the fish monitoring work group is operating right now, any task team, we don't want to create an excess burden of more meetings for all of us to attend. So a task team lead is encouraged to mainly rely on virtual meetings, and we can leverage the PNAP Teams platform for that, or if a task doesn't require any meetings, you could just rely mainly on sharing a document through the PNAP Fish Monitoring Work Group Teams channel so that you can jointly collaboratively work on a product that way, and then minimizing the actual meetings. If there's a need for an in-person meeting, we can discuss that with the core team and then as appropriate handle that, but we're focused on producing products, and the most efficient way to get to that product is what we encourage the task team lead to implement as an approach. The last responsibility of the task team lead is when a product has been successfully developed by the task team. We expect the task team lead to come back to the fish monitoring work group core team and present that product to us, and then also help us deliver that product to the appropriate group so that they can leverage it and benefit from what was developed by the task team. For task team members, again, we hope the fish monitoring work group general members who help identify topics of interest, are excited about them, will volunteer, just like we got a series of volunteers for the active task teams that we've already started working with. And that they are mainly responsible to help the task team lead be successful in generating that product, and to provide their expertise, provide their input so that they can help the task team lead produce a product. And similarly, just to stress again, we're not trying to overload people with meetings, so we encouraged task team members to engage virtually through shared document on the PNAP task team channel. That's a double team in that sentence. I don't want to have to say task team channel, or to attend the meetings that are being organized by the task team lead. So those are the 3 sections we just want to highlight because those are the ones that we hope that as we have quarterly meetings, as we identify tasks that need to be addressed, that have a high interest, that all of you will be willing to help either lead, co-lead, or provide you expertise by serving as a member on those task teams. Any questions related to those 3 sections of the charter, or any part of the charter you may have read in advance of this meeting? Okay, I think we're done. Next slide, Jen, and I'm handing it over to Casey.
All right, just trying to find all the right buttons here. So I'm just going to walk through the work plan and kind of talk about the development and what we have put together here. So the fish monitoring work group plan was formulated from data management topics and fish monitoring topics proposed over the years through PNAMP workshops, like that Outmigrant workshop that was held in 2019, and the fish monitoring work group workshop that we did in February, where tasks had been identified and we sort of voted on what was of highest interest and who was interested in leading. The work plan is managed by the core team, and management basically means just keeping track of what the development is, where we are in the process, and new proposed tasks will be added into this work plan. So each task that you see here has a task team lead and a fish monitoring work group core team point of contact. And the point of contact will facilitate and help make sure the task is progressing as reasonably as possible. And progress on these tasks will be presented to the core team during monthly meetings by this point of contact person. And we didn't list the point of contact on here. We just listed the team leads for right now. And initiation of tasks to address a proposed topic will be assessed, by the core team. And initiation will be based on, sort of this regional interest or need, like we did this voting at the last work group meeting. Also confirmation, for a task to be initiated, we need a task team lead and interested parties to help with the product development. So these 15 tasks we see here have been proposed, and some of them are starting. Many of them have task team leads, and those ones down at the bottom don't currently have a team lead yet, so they're on hold. Once a task has been initiated, like Nancy was saying, there'll be a wiki document, and product folders will be created in Teams for everybody to access from a central location. And then when the task team completes its deliverable and the task is deemed completed, again, like Nancy had said, our products will be presented to the fish monitoring work group and shared with other interested groups, like CAP or StreamNet or regional, federal, tribal, state organizations. And so the next part in this work plan is, suggestion of tasks. And so we know some of these tasks have come from workshops, but they can also come from you. And so to do that, contact a core team lead, and, we'll discuss the task. And then we also would like to use these, these work group meetings, these quarterly meetings, to weigh in and discuss with the larger group on the work plan, and what we can be adding. And so I think I've covered everything there. And so next we will have some updates from task team leads about the current tasks that are underway.
Okay, ready for the next slide? Perfect. So for this first task that we're going over, it's we ended up combining task 6 and task 7 together since they were similar in intent of trying to improve how StreamNet is displaying content on our coordinate assessment partnership Fish HLI's query. The task team lead, Tom Iverson, can't be with us today, so I'm filling in for him as the main core team contact person. So this group has been pretty active. I believe it was our first one that got started back in May. And the purpose of it was trying to improve how the coordinate assessment partnership fish high-level indicators queries. We both have a mapping one and a tabular version that is in development right now, conveyed information to the public and to the data consumers about the type of content on this fish HLI. The fish HLI currently is focused mainly on salmon, steelhead, natural origin high-level indicators, and it was originally developed to focus on high-level indicator estimates at the population scales. But as we've been growing over time, you know, the scale of the high-level indicators that are being displayed represent, for the most part, the scale at which the monitoring is occurring. So sometimes that HLI, the high-level indicators, is representing a group of population, like population going through a dam, or it might be an HLI estimate for a partial population. And we think from a bit of user testing that we can definitely improve how we convey that on our system. We also have being discussed through this task group, how to handle when a population has no information in our system, because as the general public comes or others that are interested in high-level indicators, they show up, they're looking for a population, they don't find it there, they start wondering, you know, why is it not there? So we wanted to get input on how best we can convey what's going on with that population. Is it because, you know, the HLI is coming soon, we just haven't had time or money to analyze it and produce that estimate, or is there no monitoring happening, or is the monitoring data not a good fit to inform a high-level indicator? So we wanted to have some input from a broader group, which involve all the task team members, to help us provide a recommendation that we could consider during the StreamNet steering committee and executive committees on how we can improve and how we can more accurately convey the content on the catfish high-level indicators queries. So the next step that we have right now, the team has met twice in person, once in May, which you can see on the bottom of the screen, and once in June, where they came together, they looked at the current status of the user interface for the catfish HLIs. We walked them through specific scenarios, highlighted some areas that we thought with Tom's assistance that we thought might be an issue for how people were interpreting the content, what we had heard from others in the past, as well as getting additional input from this session with the task team of other areas that needed refinement. And then from that first May meeting, Tom compiled together draft recommendations that were shared and discussed with the task team using the Teams platform and just going back and forth via email, checking in on how we need to refine them. We came back together on a virtual meeting again. None of this was in person with PNAP's help. Had another Teams meeting. We refined the draft recommendation, and now they're back out to the task team members to review and provide final input or their approval. The task team members are expected to get back to us by July 21st. At that point, we'll tighten them up, and we'll then forward those draft recommendations to StreamNet Pacific State staff, who will then prepare them for discussion during the September 2nd StreamNet Steering Committee and September 9th StreamNet Executive Committee. So we're getting close to almost being done with our first task. And that's all I have for this one. Any question before we move on to the next task? I think we're good.
So this is Russell Scranton. I'm going to jump in and present the slide for the next task, which is the CAP DES outmigrant DES update for table A4. We had a purpose and the goal and the task leads. I'll start with the task leads. Task leads are Mike Bannock and myself. We have definitely additional interested parties who have been helping this topic. The purpose was to help explain what SMOLT equivalent means and replace language in the existing DES or modify fields in the DES to help clarify data definitions. And we also wanted to discuss the DES February 2020 or 2021 survey results, specifically where people provided comments on issues or concerns related to the DES table for A4, section A4. The specific product that we're looking at is a recommendation to the CAP DDT for improvements for the definitions and fields, which would eventually go to the executive committee after confirmed from the DDT. Our next steps are that the task leads, myself and Mike, are finalizing the draft products, building on previous input received from the SMOLT workshop, the DDT survey, and we are going to be initiating the task team here very shortly once we get a couple documents clarified for sharing. Our meeting is going to be set for later this summer with a date to be selected with feedback from our volunteers and people who have volunteered based on the survey. If more people are interested in this topic, please reach out to Mike Bannock and myself. We have an initial list on the team's wiki page for this topic that identifies who's volunteered as a participant. That's it for right now. If there's any questions, please add them to the chat or speak up.
All right, for this third task, this one is being led by Van Hare and Evan Brown. I'm also the core team contact for this one right now. This one, the fish boundary names and GIS boundaries, task 5, is just about to get started. The focus of this task team is to again help StreamNet and Pacific State are benefiting a lot from this fish monitoring work group right now. To clarify how we can communicate fish population names and boundaries on our mapping tools. So in addition to salmon, steelhead, high level indicators, StreamNet also manages a lot of data sets for resident fish species and other anadromous species. And currently, we're starting to gear up a little bit more to standardize the population names that we're using for these species, as well as the boundaries that we would use to display these species on our mapping tools. The data that we house at StreamNet and Pacific State come from multiple states, tribes, and federal agencies, which generally have, all their own names and the boundaries and actual location distribution layers for their fish species. So we're hoping that through this task group, we can bring those folks together. The biologists are most familiar with these species, so we can come to agreement to what type of names, which names StreamNet Pacific State should be using on their mapping tools. So Van and Evan are currently starting to gear up for this task team. They are currently working on developing the work plan and the agenda during this month. And by the end of July, early August, anyone that has communicated interest on serving as a task member on this team, it's not too late to reach out and to be asked to be added to this task team, since we're just about to get started. All interested parties will receive a doodle poll, most likely coming from Meg, to help us schedule the first meeting, which will hopefully happen late July, early August, depending on how quickly Van and Evan can coordinate and locate a date that works for the task members. So if you're interested in joining this team, let us know. You can contact Van, Evan, or any of the core team members or Meg, and we'll make sure we'll add you to the list. And the more folks we can have helping us come together to shore up the list of fish population names and inform the boundaries that we will be using at StreamNet at Pacific State for these focal species of interest, the better it will be. And what we hope to get out of this task team is, again, a recommendation that we then will share with our StreamNet committees and discuss with interested parties at Pacific State so that we can go about implementing, figure out the best way to implement the recommendation from the task team. Any questions on this one, or if you, any volunteers that want to provide their name right now in the chat, that'd be great too. Okay. You have a question, Russell? Okay. You're muted, unfortunately.
Russ, you're muted.
Okay, in theory, I've been unmuted again. Just taking a little while on the leg. Yeah, I was just waiting to present this topic, so no comment on your previous one. Thanks, Nancy. I am assigned as the co-lead or the core team member for the carrying capacity DES task number 9. We've had initial meetings with Tim Copeland, Shuba Pandit, and Chris Beasley, but Chris Beasley has passed on this task to have Richie Carmichael step in, and we are currently setting up and finalizing some of the task definitions. So the purpose of this task is to agree to a data exchange standards for population and reach-based carrying capacity estimates. And the specific products are recommended PNAMP and partners, the CAP, XCOM, DAS specifications for carrying capacity products. This includes standards for data inputs like hydrologic networks, as well as outputs, the units of measure that these are going to be reported in. And providing definitions and fields that would be shared within these data exchange standards so people can more readily consume these data exchange outputs for carrying capacity and display them in common comparable formats. The next steps are that the task leads, we are going to be meeting later this month to finalize the draft purpose and the task description. And then we'll be initiating a meeting with all the task team participants, again, and based on people who have identified themselves as interested parties. We have, I think, over 28 people that have identified themselves as interested in this topic, and we hope to have our first meeting on this topic here late summer, early fall, based on getting the right people engaged with all the other tasks we're working on. Any questions or comments? Let's go ahead to the next slide.
I'm going to jump in before we introduce Toby Koch and say thank you to all the folks who have volunteered to step up and be part of those first 4 tasks. It's we're just getting started. Those are ideas that were kicked around over a variety of years at workshops and meetings and whatnot. And so we're excited to have those get started. We hope it generates ideas for additional tasks. So don't feel like the door is closed. We're just getting started. So thanks for indulging me, Marika. I'm done.
Okay, so it is my pleasure to introduce our Tech Talk speaker for today, and that is Toby Koch. Toby is a fellow Vandals, graduated from the University of Idaho with his master's and a BA from Pacific Lutheran University in Tacoma, Washington. Toby is a research fish biologist at the USGS Columbia River Research Laboratory in Cook, Washington. His primary research interests include evaluation of dam passage and survival, assessment of fish collection devices, development of fish passage options at high-head dams, and reintroduction of salmon and steelhead to areas blocked by dams. He also works on the effects of climate change on salmon ecology and dam management. He joined the USGS in 1998 and has since been working on fishery studies focused on Pacific salmon and dams in the Pacific Northwest. He's worked on various projects, including the use of telemetry systems to monitor juvenile and adult salmon and steelhead. So we're excited to be hearing his talk today, where he will be explaining some use of temperature sensing radio transmitters to evaluate migration success and water temperature exposure of adult sockeye salmon in the Yakima River in Washington. Take it away, Toby. Thank you.
All right, I forgot to unmute myself. All right, are you guys seeing the cover slide here?
Yep, looking good, Toby.
Okay, great. So yeah, we've been working on adult sockeye behavior and movement in the Yakima River since I guess about 2017. Some of the earlier years of that study were focused upstream of Rosa Dam, which I'll show you a map here in a minute, so you have a kind of a lay of the land in your mind. But you know, I'm not going to get into those studies today. I'm going to focus more specifically on studies conducted from 2019 to present in the lower Yakima River near the Yakima River mouth and in the Columbia River. And we'll talk to you about how we're using temperature sensing radio transmitters to assess really a variety of things in the Yakima. And so just a couple things to point out here. In the Yakima River Basin map, you can see that it occupies a large portion of Washington State. And you know, I just want to point out here at the front end, there's several large reservoirs in the headwaters of the Yakima Basin, and those are, you know, located up in the Cascade Mountain Range. And so the Yakima is a really diverse suite of habitats that goes from these high head reservoirs in the Cascade Mountains, and I would say that really the lower half of the Yakima, starting not too far upstream from the city of Yakima, is when the river slows down and really flows through an arid desert type environment. And so a lot of what I'm going to be talking about today is going to be downstream of Yakima. And so Yakima Nation began leading the efforts to reintroduce sockeye in the upper Yakima Basin in 2009. Sockeye were a native species to these high elevation reservoirs in the headwaters. But when the high head dams went in the early 1900s, it basically extirpated sockeye from the basin. And so sockeye were not present for about 100 years in the Yakima Basin, until Yakima Nation began reintroducing them in 2009. This reintroduction effort is being run basically using donor stocks from the Okanagan and the Wenatchee rivers. They're collected at Priest Rapids Dam. So as these sockeye come up the main stem Columbia and try passing Priest Rapid, they're put in a truck and taken over to Cle Elum Reservoir, where they're released. It's a pretty substantial program, and the transport numbers are based on the total run size in the Columbia each year, but they can transport up to 10,000 adults from Priest Rapids to Cle Elum every year. And there's been several years where the run's been large enough for them to do that. So, you know, a pretty substantial outplanting effort has been underway since the program began. The program has been successful at producing recruits to the Yakima River, and so adults that spawn upstream of Cle Elum Dam produce juveniles that outmigrate, go to the ocean, and are returning to the Yakima River. And so those fish, as they come back up the Columbia River, turn left into the Yakima, and there's a couple key locations that I want to point out, you know, call your attention to. The first is Prosser Dam here. It's probably a third of the way upstream to Rosa Dam. So Prosser is the first location where we can really get information on returning adults. They have dam counts there, so we know how many adults are entering the river. There's some pit tag arrays. We can get some information on pit tag fish there. And then these fish volitionally pass Prosser and move upstream to Rosa Dam, where again they're collected, put in a truck, and taken upstream to Cle Elum. So there's really the reintroduction effort now is relying on 3 stocks: Okanagan and Wenatchee fish coming over from the Columbia, and Yakima River fish coming back up the Yakima. This reintroduction effort is, the Bureau of Reclamation and the Washington Department of Ecology have been working on providing and developing fish passage at Cle Elum Dam, and this is a very large project. You can see the schematic of the dam and reservoir here. You can see this fish passage helix that's going in. There's going to be adult collection facilities developed, and this is a shot looking down into the helix. I'll call your attention to a tractor and a little cart here. This is a very substantial structure, the helix is, and there are millions and millions and millions of dollars going into developing fish passage at Cle Elum. The long-term goal is to eventually reintroduce sockeye to all of the high elevation large reservoirs in the Yakima and to develop fish passage at those reservoirs. And so there, the financial implications are substantial. There is obviously a lot of interest in seeing that this sockeye reintroduction is successful. And so that's really what's fueling a lot of the interest in our research and why we're focused down at the mouth of the Yakima, which I'll tell you more about. And so the first thing I just wanted to cue you into is this is run timing for adult sockeye at McNary Dam. McNary Dam's 30 or 40 miles downstream of the Yakima mouth, and so really the main thing to understand is that sockeye are approaching the mouth of the Yakima. They're passing McNary Dam primarily in the second half of June and the first half of July. And you can see that the run at McNary is peaking in the last week of June and the first week of July. That's pretty significant because if those fish, are available to enter the Yakima in late June and early July, there's some water temperature concerns in the Yakima, and so we're showing a USGS water temperature graph from 2018 to 2021. And when you see these peak water temperatures in each year, you see that they're occurring in late June and early July. And so these fish are passing McNary Dam. They're arriving at the Yakima River mouth, but really the Yakima River is too warm for them to enter. And so if you're not familiar with adult salmon, thermal requirements, anytime you get up 18, 19, 20 degrees, you're really getting in kind of the upper limit of where they're comfortable and when it's safe for them to be migrating and passing. And so you can see that the lower Yakima certainly gets up to 20 to 25 degrees every summer, and in a lot of summers it gets up in 25 to 30 degrees. This hot year that we have right now, it actually went over 30 degrees a couple weeks ago. So it's an interesting situation down there. I'm going to show you this whole graph in its entirety in the next slide, but there's a lot going on, and so I want to focus on one panel so you kind of understand all of the elements here. And so the first thing I'll call your attention to is the blue, the solid blue and the dotted black line. That is water temperature in the Yakima River, the blue line, compared to water temperature in the Columbia River near the Yakima River mouth. So you can see that there's a large discrepancy in water temperatures during the, June through basically early September period. And so what that means is, fish can hang out in the Columbia and still be relatively safe, less than 20 degrees for much of the time. But water temperatures in the Yakima certainly exceed that 20-degree threshold by a bunch. Also, for each year here, we're showing the total run timing graph for McNary Dam, so fish that are queuing up near the Yakima mouth. And then the new element that we're adding in here are the Prosser Dam counts. And so these are actually when we're seeing fish in the lower Yakima moving around. And so I want to make a few points here as I expand this to the larger graph. And that is what we tend to see taking place in the Yakima is a bimodal run timing. And so you can see here that in mid to late June of 2017, there was a push of fish when Yakima River water temperatures were cool. The Yakima gets above 20 degrees, and we don't see fish present in the lower river or moving through the lower river at that time until it cools back down in late August and September, and then you see some fish starting to show up. And so that type of bimodal behavior is generally present every year in the Yakima. In a lot of years, the greatest proportion of adult sockeye are entering the Yakima River in September when the river cools back down. You can see tend to have a lot of fish moving in at that time. The other thing to note is these fish seem to be very responsive to cool downs that occur during the summer. Every time you see a water temperature dip, or most times you see a water temperature dip in the lower Yakima, you see a group of fish respond by moving upstream and arriving at Prosser Dam. And so just some background on what's going on in the lower Yakima. This is a picture, showing the main stem Columbia with the lower Yakima, map here up to Prosser Dam, and there are some cool water inputs that flow in that region. So this is data that was collected on one day in June of 2019. The main stem Yakima River was already over 20. It was at 22.4 on that day. And there are tributaries, small streams that flow into the Yakima, and irrigation returns that are providing some cool water inputs. You can see anywhere from about 15.8 to 18 degrees. So pretty substantial decrease in water temperature for these tributary inputs. They don't have a long-lasting effect in the main stem, but they do provide these isolated pockets of cool water. And so there's been some interest in how fish could be using those. Basically, I think the term has been, as kind of lily pads of safety to go perhaps conceptually, from one cool patch to the next and move upstream. But there hasn't been a lot of information until our study to document whether that's occurring or not. And so in our study since 2019, we've been using temperature sensing radio transmitters. You can see a photograph of one here. Basically, these transmitters do two things. They emit a signal so which individual fish is at a specific location in time, as long as you have a receiver to detect it, so we can track movements of individuals. When you recover the transmitter, you can download the thermal profile or the thermal history of that fish while the transmitter was inside of it. And so, this study that's shown here is 2008. I think we actually did the work in 2004, 2005. It was at where the Clearwater flows into the Snake River up near Clarkston, Washington. It's a complex thermal environment, but you get really good data. These are thermal tracks from individual fish. You get really good data and records on what the thermal exposure or thermal history is for individual fish. And so, having used this technology in the past, we propose to use it in the Yakima River. And so we've been doing this study now. Basically, all the thumbtacks on this study area map are locations where we have fixed monitoring sites. And so we have a couple out here in the Columbia River at the Blue Bridge near Kennewick, up in the lower part of the Hanford Reach near Johnson Island. Keep those sites in mind because I'll talk about those when we get into the results. We have several sites in the lower Yakima River near these cool water inputs to see what fish are doing at those locations. And then we have sporadic monitoring upstream to Rosa Dam to determine migration success to where fish are collected. We've been tagging fish and releasing them at Prosser Dam, and collecting and tagging them and releasing them at the Yakima River mouth. So we have a couple different groups of fish going in at different locations to understand what's happening down there. And so the first data I'll talk to you about is data for a group of fish that were tagged up in the Yakima River at Prosser Dam. They were tagged in late June of 2019 when water temperatures had dipped down to around 20 degrees. So if you think back to that graph I showed earlier, a group of fish moved upstream in the Yakima during this cool down period. We collected a bunch at Prosser, we tagged them, we released them downstream near the mouth, and they began moving upstream to Prosser Water temperatures increased, basically during the week after we released them, and all of those fish turned around and were detected as they left the Yakima River in about 5 or 6 day period. And so on this increasing thermal limb, all of those fish left the Yakima. You'll see that Yakima River temperatures stayed above 22 degrees until late July that summer. This graph picks up in early August, so here we are above 24. And the water temperatures gradually cooled down. As they got down around 20 degrees, we started seeing fish, this is individual numbers of fish, moving back into the lower Yakima. And so again, this just supports the idea that there's a thermal barrier in the lower Yakima, and that when temperature gets much above 20 degrees, fish leave. When it cools down towards 20, they begin entering again. And so I'm going to show this figure to you a couple different times. When I talk about it on this slide, I'm just going to focus on this left half of the graph. A couple things to point out here. And so this is a map of the Yakima River mouth. This area I'm circling here right at the, where the river flows into the Columbia is where WDF&W has been using volunteer anglers to collect fish at this location. We've been tagging them and tracking them from that location. It seems to be an important point for Yakima River fish. They seem to congregate there. I'll talk about that more in a few slides. But basically, as you look at this graph, the black line is the thermal experience of the fish. So this fish, this is the thermal experience of that fish over the time that we tracked it. The blue line shows the Columbia River temperatures at the time of tracking. The red line shows the Yakima River temperatures. And again, if we focus on where this fish was detected during August of this year, it was detected at the mouth, again near Richland, Washington. It moved upstream to the Hanford Reach at Johnson Island. It came back down to Kennewick at the Blue Bridge by Tri-Cities, moved back up to the Hanford Reach. And so this type of information, we have other fish with similar detection histories, shows us that these fish are just milling around. They're spending time basically pacing back and forth, waiting for thermal conditions in the lower Yakima to improve to the point where they can move upstream. Again, this is that collection location. This is data from 2020. We collected fish in June, July, and August. You can see the number of fish that were collected and tagged. So these are with pit tags. And we looked at where did those fish ultimately end up. And you can see that the largest group of fish in each of those tagging months ended up in the Yakima River. And so that tells us that Yakima River fish are present at the Yakima mouth during all portions of the summer. There's a large percentage of Yakima River fish there throughout the summer that are waiting. And so we also took DNA samples of these fish, and we're just getting the results back on those, but it looks like, based on genetic analysis, some of these fish that ended up going upstream of Priest Rapids Dam were in fact Yakima River fish. And so I think that's some evidence that suggests that this thermal block actually contributes to some straying for this population as well. And so overall now, our study seems to show that fish are hanging out the Yakima mouth. They're milling around. They're waiting to go up the Yakima. And so what do we know about migration success? What proportion of the run actually makes it up to Rosa Dam and are transported each year? And so there's some pretty good information out there to tell us that. John Hess with CRITFIC did a study where they were DNA sampling fish at Bonneville Dam. They were also pit tagging some of those fish, and his estimate for this is a 21 report. This is actually from 2020, so this isn't quite right. But he estimated that almost 12,000 Yakima River sockeye passed Bonneville Dam. Now, the total number of fish collected at Rosa Dam last summer was only about 4,400. So this information suggests that only about a third of the population that was available at Bonneville was able to successfully transit upstream to Rosa. Some of those pit fish were also pit tagged. So 31 of these 12,000 fish were pit tagged at Bonneville. we saw that 27 of them were detected at McNary, so really good conversion, you know, through the Columbia from Bonneville to McNary, but only 4 of those fish were ever detected in the Yakima River. And so again, you know, some of this information is suggesting that a large proportion of the Yakima River returning population aren't able or aren't successfully making it to Rosa. If you look at fish that we've tagged since 2019, you can see that a really low proportion, only 1 of 60 fish in 2019 and 21 of 144 fish were successfully moved upstream to Rosa. And so again, these results from these different studies are telling us the same thing, that a big chunk of the Yakima River sockeye population is not successfully making it back to Rosa. And again, I talked a little bit about the straying. We definitely see that with some of our tagged fish showing up at other locations. And so now this is the same plot you looked at. We had this fish milling out in the Columbia for a while during the entire month of August. Yakima River water temperatures cooled down, and now we see that fish starting to move upstream. And so this is telling us a story. We see that fish detected as he moves upstream. He gets up, he passes over the first diversion dam, Wannawish Dam. You know, he's detected at Chandler Outfall. He gets up to Prosser Dam, and he's at Prosser Dam for several days. He spends a lot of time there. This is mid-September. We're not too far from the sockeye spawning period at this point. He does not pass Prosser Dam, and then we see the fish begin moving downstream, and we later recovered the tag from what we assumed was the fish had died about halfway back to the Yakima River mouth. And so this is the type of information that we repeatedly see in fish that enter the Yakima in September. We see fish move upstream, they get to Prosser, and we see tagged fish turn around and eventually leave. We also see a fair number of fish passing over Prosser Dam, but ultimately never making it to Rosa, just based on pit tag detections. And so, you know, you've got to assume, I think, that these fish are getting tired. They don't have energy reserves really left at the, you know, by early to mid-September when the Yakima cools, and so seems to be having an effect on migration success to Rosa. We talked a little bit about the importance of cool water sites, or, you know, how are fish using cool water sites. And so I broke our detections down here into day and nighttime detections. Did they arrive during the day? Did they arrive during the night? This is from 2019 and 2020. And I shaded in gray the sites that are known cool water sites. And so you can see Sunnyside Dam here, you know, not a known cool water site, data for 19 and 20. I think the thing that jumps off the page here to me is that when you look at the cool water sites, you see that there's very low residence time. The majority of the fish tend to spend a few minutes, a couple hours at near the cool water sites before they move upstream. But when you start looking at the diversion dams, so Wannawish Dam, Prosser Dam, Sunnyside Dam, that's where we tend to see our largest residence time. And what that tells me is that there's some migration delay that's occurring. Those fish encounter the diversion dams, they don't find, you know, a really attractive passage route, and so they're probably wasting time and resources at these locations. This is some video that we collected last year. This is at Wannawish Dam. The video is going to show a single sockeye jumping up there. You can see it right in this lower corner. I'll show it to you one more time. You can see the single fish. It's trying to actually climb over, you know, jump over the dam versus using the ladders at the dam. The reason we collected this video is we actually observed at that time several fish trying to do this. And so I think what this suggests to me is that there's definitely some passage challenges at these Yakima River diversion dams. And, you know, that's probably what's leading to some of these extended residence times. The other thing that we're starting to get a handle on is some predation that's occurring from birds, you know, avian predators in the main stem Yakima. White pelicans in particular are pretty common in the Yakima River. Here you can see a bunch of them actually just sitting on rocks, swimming around. This is at the Prosser Outfall, so this is a place where juveniles re-enter the river. But what we started seeing at Prosser Dam when we were tagging in 2019 is a lot of these injuries, these dorsal injuries, You know, these are fish that we hadn't handled before. We just collected them for tagging. But, you know, when we started seeing these, it seemed to me like, you know, how would you explain that? That sure seems like, you know, some kind of a bird injury, or it could be explained by a bird injury where, you know, a large white pelican is taking swipes at these fish as they swim by. And so you see this common injury showing up at Prosser. And in fact, the temperature sensing radio transmitters really helped us to document this further in 2020. So again, on this graph, you see Yakima temps, Columbia temps. This fish we released at Prosser Dam in the forebay, so it moved upstream towards Marion Drain and began moving upstream towards Rosa. You see it, you know, experiencing some water, some different thermal conditions as it moves upstream. You get to this point on the 23rd of June, and you know something is going on here, right? This temperature spikes up to 34 degrees. That is the peak of the water temperature that can be recorded by these transmitters, and that tag remains at this peak for almost a day or just over a day. So what's going on there? The body temperature of American white pelicans is about 40 degrees, and so, you know, I think to me this is pretty indicative of a fish being consumed by a white pelican. The tag is in the fish for this period of time. The fish, the bird passes the tag and drops it near the river. You know, we recover it a few days later. When we go back and look at 13 fish that were released at Prosser Dam in 2020, we have 6 fish that show this exact thermal profile. And so I think that tells me, you know, that avian predation is occurring to some degree in the Yakima. I'll also note that some of our pit tag only fish that were released at the Yakima River mouth were later, those pit tags were later recovered on bird colonies out in the Columbia. And so, you know, there's some indication that this is happening. Now, an adult sockeye is a big fish. You know, obviously a lot, you know, I think everyone on this call recognizes that the avian predators can eat smolts, no problem. I just wanted to point out your attention. This is a study that appeared in 2016 from suckers in the Upper Klamath River Basin. They were documenting water bird predation. And, you know, this sentence really grabbed my attention. Tag suckers ranging in fork length from 72 to 694 millimeters were consumed by white pelicans or double crested cormorants. The largest sucker that was consumed during the study was a female that was 730 millimeters long. So that's about 28 inches. So that's, you know, just about sockeye size. So I don't think, you know, I think that's just further justification that these birds are capable of eating, you know, adult sockeye. And so in summary, you know, what do we know? What have we learned? I think the temperature sensing radio transmitters have provided us, you know, with some new insights, the ability to collect some new information. We can track we can track movements of individual fish. We're able to look at residence times both near cool water sites and you know, at diversion dams. We can document the thermal experiences of tag fish. You know, with this presentation, I didn't get into, you know, talking about, you know, biological implications of being in 20, 22, 24 degree temperatures. There's obviously that element to the data set that we can get in, and ultimately we're able to start looking at, you know, determining migration success in the Yakima River or identifying factors that are contributing to migration failure. Many of our Yakima River sockeye were documented holding in the Yakima mouth. They seem to mill in the Columbia waiting for temperatures to cool. The data, at least at this point, don't suggest that those cool water inputs in the Yakima are, you know, that fish are spending a lot of time at them. So I think, you know, environmentally, those cool water inputs probably, you know, are important for the lower Yakima, but I don't know that they're necessarily providing a big benefit to the migrating sockeye. Certainly, I think we're showing that there's evidence for avian predation and some passage issues at Yakima River dams. And so that's what we know to date. We had started down the path here in the last couple weeks of doing a follow-on study in 2021. A couple things have led to that research being suspended. The first is The run prediction for sockeye to the Yakima River this year was going to be very, very low. Cle Elum Reservoir did not fill in 2019, and therefore sockeye smolts were unable to get out of Cle Elum Reservoir, and so the adults that would be coming back from that, from that lack of production in 2019 were anticipated to be very, very low. And then obviously, I think you're probably all aware that we're dealing with a really hot summer right now. And so water temperatures in the Yakima are over 30 degrees. We've had a couple collection efforts at the Yakima River mouth, and we're either not collecting fish or the fish we're collecting are not in good condition. And so we basically just suspended the study for 21. And we're looking ahead to 2022 and certainly hoping for, a larger adult return to the Yakima and cooler waters. And so with that, I will take any questions.
All right, thanks, Toby. That was fantastic. I felt like it was really interesting to see the bimodal run timing and the ability to identify the thermal barrier in the Yakima. And so insights on temperature-based movements that always fascinates me. So if anybody else has any questions, feel free to unmute yourself. That's the top, usually the top with the little microphone, or you can type your questions into the chat. We'll give you guys a little time to do that. All right, it looks like Jeff has a question. Would it be possible to get a pit tag array in the lower Yakima River to determine whether sockeye mortality is occurring in the lower Yakima or sockeye are not entering the Yakima?
Yeah, that's a great question. That's something a lot of the pit tag arrays in the Yakima River, Yakima Nation has been putting those in place. Either with internal funding or with funding from the Bureau of Reclamation or Department of Ecology. And so that is, I totally agree with your observation. We agree with that observation. And so that's, long-term we're managers, I think, are thinking about that. And so, it's there's a lot of public access to the lower Yakima River, and so concerns with vandalism and damage are obvious, and there's some big flows that come out of the Yakima during certain portions of the year. So there's some of the typical challenges with getting a pit tag array in place, but managers are definitely thinking about that, and that's on our radar.
All right, excellent. Great question, Jeff. Next is Brady Allen. He's asking, how did you collect the fish at the mouth of the Yakima?
Yeah, so Washington Department of Fish and Wildlife has been organizing volunteer angler teams to help us. And so the volunteer angler teams are going out 2, 3, 4 times a year. So we'll identify dates a few months in advance. We go out. We have a tagging boat on site. There's 5 or 6 angling boats out there. They collect fish. We pull up, take the fish, tag it, drop it back in the river, and it's on its way.
All right, excellent. And it looks like Devin, you have your hand raised. You can go ahead and ask your question.
Yeah, thanks. Great presentation. So we did a radio tagging study the last 2 years here in the Nooksack River. My question for you is, how did you recover those tags?
Yeah, great question. So some of those tags, if a fish comes back to Rosa Dam and it's got a transmitter, before they load it on a fish truck, they're pulling the tag out and saving them for us. A lot of our fish that are dying en route, either in river or, via bird predation, when these tags are out of water, they have a very large read range. You can hear it, a mile away or something. And so we have mobile trackers who are out, monitoring fish during the study. When they see a tag, when they see a transmitter is at a certain location for an extended period of time, we'll float the river, do what we have to get there and recover the tag. And so that, I would say that that's the majority of our recoveries have been via mobile tracking.
Cool, thanks.
Awesome, very cool. Technology can be great. Shuba, I see your hands raised. Go ahead and ask your question.
Hey, Toby, it's a great presentation. So lots of good information for us. So I have a one just a curiosity that the predator mortality is a very big issue in Columbia River. So it ranges from 5 to 25%. So how about, have you done any analysis how what percents are contributing to for the reducing the survival probability for this species in Columbia in Yakima Basin? Or basically what the percentage of mortality from the predator, from the avian predation?
Yeah, so Shuba, if I'm understanding, how much avian predation has been documented out in the Columbia versus in the Yakima? Is that what you're asking?
Kind of, yes.
Yeah, so that's a great question. Can you guys, are you looking back at the PowerPoint now? Can you see that?
Yeah, we can see that.
Okay, so, whoop. So these data are basically, I think these data in this, go into this graph are only from, pit tag recoveries. And so the percent of pit tags that have been recovered from fish out in the Columbia River is fairly small, 3 to 5% based on fish that were collected last July and August. And so, a relatively small percentage. I think the thing to keep in mind is, for us to get information on that group of fish, basically the bird has to consume the fish and tag. It has to go to a known bird island. It has to pass the tag at that location. Crews have to sample it and detect the tag there. And so I suspect that the percent of avian predation that's showing up in this graph is, biased fairly low. In other words, probably more sockeye are being consumed than are showing up in this graph, and tags are being deposited in other areas as fish are spending their time, around the basin. Did that answer your question?
Yes, good. Thank you, Toby.
You bet.
All right, excellent. All right. We're going to take one more question. It is, Marissa's curious if the tags are sensitive enough to detect small-scale thermal refuges like pools or seeps.
Yes, definitely. And so kind of two-part answer to that question. They're sensitive, their sensitivity is less than 0.5 degree C. And so we see the tags providing information on very small scale changes in water temperature. We also see them responding fairly quickly. In other words, one of the questions we had initially is if a tag is inside a fish and a fish moves into a small patch of cool water, how fast will that tag respond? And so we've done some tests with that, and it looks like it only takes, a minute or two for the tag to pick up and actually record that temperature change. And so, yes, they are sensitive enough, and they seem to be responsive enough inside a fish to provide useful information.
All right, excellent, excellent. And we'll be sure, as long as you're okay with it, Toby, I can include your email address in the post follow-up stuff in case anyone wants to contact you and kind of pick your brain a little bit.
You bet.
All right, excellent. Well, thank you so much, Toby. That was awesome.
You bet. Thanks for the invite.
Yeah. And then you can just click that X and then Jen will be able to share her screen again. All right, perfect. So we are stepping into our wrap up. I'm going to start off and then I'm going to kind of hand it over to Russ to talk about a shared publications thing that we're kind of kicking around. As you can see from the slide, these are the upcoming tasks. We try to stay a little bit flexible with everyone's schedule. So this is the stuff that's going to start in the fall, the CAP DES, StreamNet related data DES, juvenile density DES, rotary screw trap operation DES. And then one more thing that I'd like to mention is if you're new to this group and would like to make sure you're getting information about the meetings and the follow-ups and everything, feel free to join our listserv. I'll also provide a bunch of links in the post-meeting follow-up. This meeting is also being recorded, so in case you want to watch it again or forward it on to a coworker, all of this stuff will be available after the meeting. Hopefully I can get that out this week or next week. And always feel free to email me if you have any questions or you want to get connected to anybody. So I'll hand this over to you, Russ, to talk about the paper.
Thanks, Meg. I'll try to jump in here and share my screen as well. Let's see if this works here. Are you guys able to see the wiki?
Yep.
Yes.
Yeah. Just really quickly, we talked about it at our first meeting in February. One of the ideas we kicked off was being able to share new publications. We created a wiki page that identified, a specific publication. So if you click on if you access and join the Microsoft Teams, the idea is that under the general team, we have a wiki page. In the first section we have is information sharing, papers and findings. Previously, we presented and shared these two publications, but since our last meeting, right after it, we had the emerging technologies workshops and presentations. From that, IDF&G shared with us a parentage-based tagging, improves escapement estimates, is ESA listed adult Chinook, salmon, and steelhead in the Snake River Basin. This paper was recently published and shared, and we've posted it here on the wiki page for people to be able to access. I think there's a lot of interest given a recent council presentation on publications, on research. So the more opportunities we have to continue to update and share peer-reviewed publications as they come available, that might be relevant to our community. We're hoping to see if this is a method that may work, or working with the CRITVIC, Columbia Basin Library, and other entities are possible solutions. So please feel free to log in, add publications as they come out, and provide links if available so people can actually read them. So that's my update on the wiki page. So thank you.
All right, and then Jen, if you're gonna pull up the adjourn slide. And when I send out that information, I'll give you guys a link and all the information to be able to join the team. We're still kind of working on making it accessible to everybody. So I just want to say thanks to everybody for joining today and all the work that everybody's been putting in on this project. We're doing quarterly meetings, so every third Thursday. So our next meeting will be October 21st from 10 to 11:30 Pacific. So we look forward to having you guys join us, and I appreciate you showing up today.